the electrical activities of pyramidal cells in the weakly electric fish. He's doing a great job. He has one paper published and 2 papers under review. And he's looking for a postdoctoral position. So if someone is recruiting, just let him know. Yeah, I'm looking forward to hearing your talk.
So hello, everyone, and thanks for being here today. It's a pleasure to be here with you and meet you in person. As Anna mentioned, I'm a PhD student at the Department of Physiology at McGill University. And today I will talk about the longest standing debate in neuroscience about how neurons encode information and how that information is interpreted in their electrical activity. Specifically, I will walk you through how burst firing happens in vivo and how it shapes information encoding across neural population, all within the context of invariant coding in sensory system. So to begin with, what does invariant coding even means? To give you an example, let's look at the pictures of poodle dogs versus teddy bears here. I think you can all find the dogs in these pictures, irrespective of their differences in context, in their gesture and lighting condition. This refers to the ability of our brain to perform context-independent object recognition, known as invariant coding. And this ability enables animals and species to find food, to detect threats, and make communication with each other, and it's very important for the survival of the living organism. Perhaps a good example of invariant responding neuron was found in the pioneering work of Koirova et al. At UCLA and Caltech in 2005, where they discovered a specific neuron in the inferior temporal cortex that exclusively activated in response to different pictures of Jennifer Aniston, the actress, while it was mainly inactive in response to other celebrity pictures or other objects. Additionally, they also found other neurons that respond only to other objects, for example, Hail Berry or Eiffel Tower, while they remain inactive in response to other objects. However, despite the interesting finding they had, we know that the inferior temporal cortex is located in the higher levels of visual sensory processing pathway. But we know that the sensory information is presented way earlier through the retina. So one of the open questions in system neuroscience is that how this information is passed along the successive brain areas, and how does it eventually lead to perception and behavior of invariant information. To understand how the brain performs invariant representation of sensory input, we must first determine how neurons process incoming information in the brain. This process is known as neural coding, and historically, studying neural coding has been challenging mainly because of 3 reasons. First, we know that the individual neurons display large heterogeneities in their spiking activity, even in response to a common stimuli, and that such a heterogeneity, the role of such heterogeneity remains largely unknown. Secondly, we know that neural activities are often highly patterned. For example, some neurons are showing the tendency to fire clusters of action potential in response to a stimuli, while sometimes they show single action potential to the same stimuli, known as tonic firing or burst firing. And finally, we know that perception and behavior in the nervous system are not solely dictated by the activity of individual neurons. Instead, the incoming information in the brain is processed by the combined activity of large neural populations interacting with each other in a collective manner. So to study population coding in sensory system, we need a good animal model to study this behavior. Therefore, we use the gymnotiform wave-type weakly electric fish. This species is ideal because of its well-characterized anatomy and physiology. It essentially can generate an electric organ discharge known as EOD through their tail. This EOD has a quasi-sinusoidal waveform which can be used to scan the environment. It is important to notice that this EOD only acts as a radar, and it's the interference of any object with the EOD that allow the fish to detect objects and make communication with their conspecific. When the two fish are near each other, the interference between their EOD causes sinusoidal amplitude modulation with an envelope frequency equal to the difference between the two EOD frequencies that we know as beat. And that's a signal that tells the fish that there is a conspecific nearby. These fish detect the sensory input through electroreceptor afferents located on the animal's skin and project that to the electrosensory lateral line lobe. It's the first stage of sensory processing unit in the electrosensory system. The pyramidal cells within the ELL receive this input, and after some processing, project that to midbrain areas known as torus semicircularis and nuclei provincialis, and through there to higher brain area. They also receive feedback from midbrain, from nuclei provincialis and AGP that regulates their spiking activity. It's been shown that in vitro studies that these ELL pyramidal cells generate a specific type of chaotic burst firing patterns in the range of gamma frequency oscillation, typically in the range of 20 to 80 hertz. And this bursting phenomena is known as ghost bursting. This burst mechanism is essentially based on a ping-pong interaction between the soma and the dendrite in the cell. So when there is a somatic action potential, it backpropagates to the dendrite, causing an action potential in the dendrite, which propagates to soma. And this interaction continues until the last two spikes in the soma, the period between the timing between the last two spikes in the soma becomes shorter than the refractory period in the dendrite. So there's an action potential in the soma, but in response, dendrite remains mainly silent, and that terminates the burst. However, after we perform recording in vivo from these cells, we see that this bursting doesn't appear in vivo recording. So mainly the bursting in vivo are usually shorter, have shorter period, typically 2-3 spikes per burst, with no sign of this depolarization after potential or dendritic failure or burst after hyperpolarization. And the experimental studies done in Chakram labs and others showed that the SK channel activation due to the calcium calcium release through either NMDA or buffering is the main reason for such a differences between in vivo and in vitro burst firing. So we believe that computational modeling can help us to explain the activity of these ELL pyramidal cells in vivo. Therefore, our first goal was to develop a computational model to capture their in vivo spiking activity of these ELL pyramidal cells, especially their intrinsic dynamics of burst firing. So to do that, we recorded intracellularly in vivo from a population of ELL pyramidal cells. And as you can see from this recording, their spiking activity looks a lot more noisier and more varied compared to what we typically see in vitro. These neurons can generate sometimes just a single action potential, or as I mentioned, a cluster of bursts in vivo. And if we take the time difference between these consecutive spikes and plot the histogram of the interspike interval distribution of these spike timings, we might see a bimodal distribution and similar to this data. And if we set a threshold, we can separate these two modes of their spiking patterns. So if we set this threshold, we can essentially separate the spiking activity into bursts and isolated spikes based on the timing of the spiking activity. This is what we see from the data. So now we want to model this. To develop a model, we coupled the revised conductance-based Hodgkin-Huxley model that was initially designed for in vitro spiking activity of these ELL pyramidal cells by Doiron. And we used a flux balance based model that describes the calcium immobilization in pyramidal cells. This model primarily includes the sodium channel, delayed rectifying potassium channel, and leak current in the soma. In the dendritic compartment, we also have the sodium, potassium, delayed rectifying potassium, and leak current. But we also included the SK channel and NMDA current in the dendrite. Because as I mentioned, it was shown that SK and activity of calcium activity are important factors for replicating that in vivo spiking activity. I have to mention here that in the soma, we also have expression of SK and NMDA, but the level of expression of these channels in the soma is not significant. So we just use that approximation and excluded that from the model just for simplicity. Then these two models are coupled together. These two compartments are coupled together through a resistive coupling. In order to model the NMDA, we use the 5-state Markov model borrowed from the Destesh model for explaining the NMDA current. Here, it's a Markov model, and O shows the open probability state of the NMDA receptor that eventually leads to the calcium entry into the cell. And finally, to model the calcium dynamics, we utilize the Hodgkin-Huxley formalism and incorporated PMCA, SERCA, and IP3 receptors across the fluxes across the cell membrane and the endoplasmic reticulum. So now that we have the model, we can essentially use a bunch of parameter values and simulate this model. From the simulation of the model, we can get the membrane potential traces, and then we can extract the interspike interval distribution from the model. Similarly, we can from the recording data, we can perform a spike sorting and extract the ISI distribution from the data. And the goal of this optimization is to minimize the error between the ISI distribution we get from the data and the model that can guide the parameter optimization, and we can find the best set of parameters that can explain this spiking activity. If we find the best set of parameters for each individual cell, as I showed you the spike train of the recording earlier. You can see here for this 20 example ELO pyramidal cells that we obtained from the simulation of the model. You can see that the model does a good job to essentially get this heterogeneous spiking activity as well as the bursting dynamics we see from in vivo activity of these ELO pyramidal cells. And you can see that the goodness of the fit with respect to the data is almost acceptable. So the model can do a good job to replicate this spiking activity of these ELO pyramidal cells. Next, because it's this biphysical model is a complicated model and has lots of parameters and lots of currents, we try to see if we can use a phenomenological model, a simplified model, to essentially reproduce the same spiking activity of these ELO pyramidal cells in vivo. This can be beneficial because this phenomenological model can be used further to study the population dynamics and couple these neurons together and study the network behavior. So therefore, we use this Hindmarsh-Rose formalism we use this the Hindmarsh-Rose model for creating a model that can generate this in vivo spiking activity of the ELO pyramidal cells. Originally, the Hindmarsh-Rose model cannot generate this increasing frequency burst clusters, so we have to modify it. To do so, we modified the Hindmarsh-Rose model by adding a slow adaptation variable that provides feedback to the first variable, which corresponds to membrane potential activity. And if we add the stochastic input to this model, we can see that the model does a good job to generate all different statistics that we obtain from the spiking activity of these ELO pyramidal cells, including the mean firing rate, the fraction of bursting in these cells, the mean ISI distribution, median, and etc. So we can see that the model can generate realistic spiking activity of these ELO pyramidal cells across the whole population. Now that we established that our model can reproduce in vivo spiking activity with high accuracy, we can use this model to explore the sources of a spiking heterogeneity within ELO pyramidal cells in vivo. So to do that, we collected extracellular recording from the spiking activity of ELO pyramidal cells using neuropixel probes, and fitted the model to a large population of recorded neurons under baseline condition, and identified the parameters that can best fit each neuron. Then we use these model parameters obtained through the fitting process to estimate the normalized distance between all cells in the data set. When we sort the pairwise distance for all model parameters based on their variability, we find that the extrinsic synaptic input, as well as the firing frequency of the presynaptic neurons, contribute primarily to the spiking heterogeneity observed in these ELO pyramidal cells, while intrinsic properties such as the conductance of the SK channel and the somatodendritic area ratio, the ratio between the soma and the dendritic compartment, play a secondary role. So we believe that the main source of spiking heterogeneity in these ELO pyramidal cells in vivo are the extrinsic sources. Then we also validated this finding by using our Hindmarsh-Rose model. So after fitting the Hindmarsh-Rose model to all these pyramidal cells, all these recorded cells, we use the sensitivity analysis to see how parameters are varied to explain the variability in the data. So for those of you who don't know what is Sobol index, it's essentially the ratio of the variance across each one of these parameters divided by the variance in the data, and those that are varied most would have higher scores, and those are the main parameters that contribute to the spiking heterogeneity. So as you can see here, the intensity of the noise and the coefficient of the inhibitory feedback for the slow adaptation variable that we added to the model play the main role, which is kind of consistent with what we see here, because here we see that the extrinsic sources and the slow variables, because SK is dominated by the activity of calcium, and calcium is a slow variable in neurons, are the main factors contributing to this heterogeneity. Then we use this Hinmarsh-Rose model again for a sanity check, and feed it with one example cell to see how this model performs with or without a stochastic input. So in these two simulations, all the
video. Sorry about that, everyone. Just 1 second. Well, go ahead, sir. Yeah, please.
Okay. So once we fitted all model parameters for control condition and after feedback plot, we can essentially compare which parameters are varied the most to explain this feedback plot condition. And we saw that the model predicts that the extrinsic synaptic input, the intensity of the extrinsic synaptic input and the firing rate of the presynaptic neurons should reduce significantly to explain the feedback plot condition, which kind of validate our finding that these are the parameters that contribute most to the heterogeneity, because after feedback plot, the variability in these cells also significantly reduces. I can show you the result later. Then for another example, we know that neuromodulation serotonin would alter the spiking activity of these pyramidal cells and increase their firing activity and variability across population. So we also applied serotonin to these cells and then record under baseline condition and after serotonin application and did the same thing. So essentially we fitted the model under baseline condition and after feedback plot, after serotonin application. and compared the variation in the parameters of the model. So we know that serotonin would increase the spiking activity by blocking SK channel. This has been shown previously in many studies. And the model, without having this information, was able to generate this spiking activity after serotonin application. And when we compare the parameter variation, we realize that the model has to reduce the conductance of the SK channel to replicate the serotonin scenario. But also at the same time, we saw that the change in the intensity of the synaptic input coefficient in the model has to increase significantly, which means that these neurons receive more synaptic input from other areas, which makes sense because serotonin generally increases the spiking activity. So more input to these cells. So far, I showed you that our computational model reproduced the burst firing and spiking heterogeneity observed in the ELL pyramidal cells. The parameter sensitivity analysis revealed that in vivo spiking activity is shaped by the interaction between the stochastic synaptic input and a slow variable such as calcium and SK. And we did the pharmacological validation of these predictions that we saw with the model. Now, next we want to merge this behavior of, we have a model that generates these patterns of spiking activity, including bursts. We have the heterogeneous spiking activity, which we don't know what's their role in population coding. Now we want to merge these two and see what is the functional role of this spiking activity in vivo. So we focused on the role of burst firing and reconcile it with the heterogeneity to the encoding of sensory input and its impact on perception and behavior in the context of invariant coding. Recall that earlier I showed you when the two fish are in close proximity, their EOD would generate a perturbation, which is shown as the envelope frequency of the beat, which is the difference between the two EOD frequencies. That's where the two animals are nearby. But if one fish decides to communicate with another, it emits a transient signal called chirp, which consists of a brief increase in the EOD frequency that occurs on top of the beat signal. So you see the increase in the frequency of the emitter fish. And the same chirp can have different features, including the same chirp can have different amplitudes, can have different duration, and can occur at any arbitrary time points, which means that can be introduced to the envelope at any arbitrary phases of the signal. So this is kind of similar to when we try to communicate with each other. We can be close to each other, but at any time point, we can decide just to talk to each other. If we play with these three features, duration, amplitude, and phase, we can have the same communication signal introduced in different ways. Kind of similar to having different representation of the same picture under different lighting condition, different gesture, and etc. So if we just look at the correlation of how these chirp signals are introduced on the beat, we can see that these signals looks very different from each other. If we just change one of these these features in the chirp signal, we can see that the chirp signal can have different shapes and they are not correlated or similar together at all. But somehow the fish is able to recognize this. And that's the invariant coding function that these fish are able to do so. So we want to study what's their mechanism. As I showed you earlier, I told you that the response of a given neuron can be highly patterned, and neurons can transfer the sensory information using these different patterns of activity. For example, they can generate just a single action potential, if you recall, or a burst of action potential in response to the same input. So if we separate our spike train here for this neural activity and separate it into the burst and isolated spike activity based on that bimodal distribution that I showed you earlier, we can see that the responses of these neurons for the burst fraction looks more similar to each other, to these two different chirp signals, compared to the responses of the isolated spikes. So we see that kind of burst is responsible for transferring this invariant information of the sensory input. If we repeat this process over multiple trials and we just plot the histogram of the simultaneous spike timing of these neurons over different trials, we can have a distribution of pre-stimulus time histogram, which is kind of similar to the firing rate of these neurons. And if we do so for these all trials of a given neuron, we can have the pre-stimulus time histogram of a single neuron, or we can just combine these spiking activities of population of neurons at the same time, and we can come up with the pre-stimulus time histogram of the population of neurons. If we do that over the network of the population of cells, we can see that the responses of the PSTH that we get for all the spikes of the neurons across the population to different chirp signals are not very similar. But if we separate the burst fraction of the same spiking activity and build these PSTHs, they become more similar to each other, as you can see in these red firing activities. But at the same time, if we look at the isolated spiking activity, they are not very similar to each other. So now let's look at the firing responses of ELL pyramidal cells for all chirp waveforms I showed you earlier, and see compare them together. So if we compare the firing rates of neurons on top panel, if we just compare the firing rates of neurons on top panel, it should be obvious to you that the burst spikes looks more similar to each other to all these different chirp waveforms compared to the isolated spikes or all the spikes. To all the spikes trains. If we estimate the correlation between all these burst spike, all the spikes or isolated spikes in response to different chirp waveforms, and just estimate the correlation, you can also see here that these responses are a lot more similar to each other compared to isolated spikes and chirp and all the spikes. And this is not what you see in the, if you take the correlation of just the chirp waveform initially. So it means that these neurons are able to generate invariant responses by the burst firing fraction of the spiking activity. This is a novel function for burst, and which means that it means that the burst firing are responsible for invariant encoding of the communication signal. Then the next question is that is it a function that you can see at the individual cell activity, or is it a population activity of these neurons? So we took these PSTHs for single neural activity across different trials, and separate it between isolated spike and burst. And as you can see, at the single cell level, they don't look very similar. And there is no difference between the all spikes, burst spikes, or isolated spike activity. But if we combine the neural activity together, you can see that the burst activity becomes more similar. At the same time, there's not much difference in the invariant responses of all spikes and isolated spikes, which means that the burst firing are responsible for invariant encoding of sensory information, but at the population level. So we really need neurons to have correlated activity and combined activity at the population level to see the responses for invariant coding. Next, we wanted to check how because we know I just showed you that these neurons have an intrinsic burst firing which relies on the somatodendritic interaction. So we wanted to check how the intrinsic properties of neurons are involved in encoding for invariance. Since this burst mechanism, we already showed that the model is able to generate this burst mechanism. We can alter the model and see how it affects the invariant encoding by playing with the burst fraction. So to do that, we just targeted the sodium channel in the dendritic compartment, because we know that if we terminate the sodium channel in the dendritic compartment, we essentially terminate the ping-pong interaction between soma and dendrite. So we don't have an active burst mechanism in the model. And this way we can make sure that there is no intrinsic burst anymore, but we can keep the firing rate of these neurons the same and just compare these cases. So if we vary the conductance of the sodium channel in the dendrite, we can see that for lower values of GNAD, the burst fraction is minimal, and similarly, the correlation between the responses to different chirp signals is also very small. But as we increase the GNAD, we get more bursting in these cells. And as the result, the correlation or the similarity between the responses to all these different chirp stimuli become more similar. But the interesting thing we see here is that there is a sweet spot for the value of burstiness in these neurons. And as you can see, if I increase the GNAD in the model, obviously we get more bursting, but then the correlation becomes smaller than this sweet spot. What it means is that these neurons are intrinsically tuned to generate just enough burst to maximize the invariance. So too much bursting is not beneficial for this invariant coding, and we just need the right amount to have the maximum invariance. This is interesting because then if we compare this burst fraction, the invariant responses, we get the model for the optimal value of GNAD. It's kind of similar to what we see in the data, which validates that these neurons are tuned to have just enough bursting. So far I showed you that the burst responses are involved, the burst firing is involved in invariant coding. I would give you a brief example of how burst firing can be involved in synchronization activity and what's the role of modulation at the population level. So we know that the burst responses, the burst firing can promote population level synchronization across the neural activity. I used here that modified Hinmarsh-Rose model and coupled these neurons together to generate the activity across the population in response to a chirp stimuli. And we know at the chirp, these neurons should generate synchronized activity to maximize the invariance. Here, I just use a random coupling of these neurons up to 200 and see that The burst firing generates the synchronized activity by increasing the firing rate. But in reality, we know that this connectivity between neurons can be adaptive through time. So what I did, there are different factors that can affect this synaptic connectivity and intensity of the presynaptic inputs these neurons receive, including the neuromodulator serotonin that I explained to you earlier. So what I did here, I added synaptic time plasticity rule and let these neurons to be coupled together, but these coupling intensities can change through time and evolve through time. When we do this, we see that at the chirp, the synchronization across the neural population would increase significantly, and these neurons becomes more synchronized, mainly due to more bursting. If you look at the adaptive weights of these signals at the chirp time, we can see that this is what you get from a fixed synaptic weights. But then if you let the neural, the network to have adaptive wave and see what it happens to the connectivity of these neurons at the chirp signal, you see that the interaction between neurons increases significantly. This is interesting because previously it was shown that after rapid stimulation, which releases serotonin to ELL pyramidal cells, the neural variability between these neurons would decrease, which means that the neurons becomes more similar to each other or more synchronized to each other. And what we see from these two different simulations with the fixed weight and adaptive synaptic weight, we measure the synchronization across the network, and we see for the adaptive weights, the normalized synchronization is kind of similar to when you make the rapid stimulation. These two effects are the opposite because this is neural variability, which is the inverse of the synchronization. So far I told you about the role of burst firing, but what about the heterogeneity and activity of isolated spikes? So we know that we have this burst firing that can sign as the detection can be used for an event detection to sign an important stimuli. But then we know that this heterogeneous activity also exists in the neural data. So what we did here, we tried to see these neurons are able to recognize that these are the same chirp signals, but they are also able to recognize that although these are the same chirp signals, there are small variations in their feature, and they are able to see that. Kind of similar to us. We can see that these pictures are the dot pictures, but we can also tell the differences in the representation of that input. So what we did here, we tried to measure the discrimination between different chirp signals using the burst spikes or isolated spikes or all spikes. So here we just build a decoder to essentially train this decoder to respond to the chirp signal and then see what's the probability of prediction of this decoder, how good this decoder can perform, can predict other chirp signals. If we do so for all the spikes, the decoder does a good job to discriminate the different chirp signals for different representation of these chirp signals, because here I have to mention that we have different trial representation of the same chirp signal. So these neurons should be able to recognize that this is the same chirp signal. It's just a different trial of the representation, but it is different from the second chirp signal and so on. So these decoders can predict the trial variability with high accuracy, which means they can discriminate between different chirp signals at all the spike level. But if we just look at the burst spikes, they are not able to recognize different chirp signals. They are not able to discriminate between chirp signals, which means that the burst spikes are not good for discrimination tasks. But if we look at isolated spikes, we can see that the isolated spikes are contributing more to discrimination of different chirp signals. So we measured this discrimination here, and as you can see, the isolated spikes are more significant to decode the discrimination, but burst signals are more involved in the invariant coding. So I showed you that burst firing enhances reliable and uniform encoding of chirp signals at the population, which supports the invariant representation. Pyramidal cells display synchronous activity during chirp by increasing their burst firing, and this synchronous activity can be modulated by modulator serotonin. And at the end, the bursting and heterogeneity in isolated spikes support a parallel coding scheme. So burst signals, burst firing promote invariance and detect important events in the brain, while variability in isolated spikes can be used to encode fine-tune details of a stimulus attributes. At the end, I want to thank my supervisors, Dr. Kadra and Dr. Shakran, and our collaborator in the lab, Dr. Metzen, who helped me to collect this data, and Dr. Bashiwan for contributing to this project. And thank you, thanks everyone in the lab, and thank you so much for listening.
Questions? Maybe there are questions also from the. Yeah, Fred.
I was curious about your use of the simplified model, the marshmallow.
Yeah.
Because I'm familiar with it. So, well, my main question is, how do you have a good mapping between your complex model and that one, or it's just an intuition that ML has been used in similar questions? Can you map the parameters or the contraction of your parameter space?
So that's a good question. So what we did for the Hinmarsh-Rose model, because it's a phenomenological model, we cannot essentially generate the details of the shape of action potential. But here we can tune this model to generate the spike timings or patterns of the spiking activity in these yellow pyramidal cells. And what is interesting for us, because we don't have a working phenomenological model for these cells generate this specific ghost bursting dynamics, which despite all other neurons that we know, they increase their frequency along the burst. And we know most of the phenomenological model that we know, including like Zikevich model, LIF, they generate decreasing frequency bursts. But we wanted a model to generate that, and we know that the interaction between the reason behind this ghost bursting dynamics is because there is a time difference in the time scale of the dendritic compartment and somatic compartment. So soma generates faster spikes, and at some point, because dendrite is in the refractory period, it terminates the burst. That's the intrinsic bursting dynamics. So that's why I added this slow adaptation to essentially generate some time scale difference between these compartments, and that's how I managed to generate this spike.
Okay.
Any other question?
Is there a question in the chat?
No, there is none.
No questions in the chat.
Yeah, go ahead.
Yeah, that model here. How are each of the parameters? Did you infer them from some known biological phenomenon or constants, or did you just machine learning your way to them?
So
from just explainability perspective and extraction, feature extraction.
Yeah. So in this model, for the three first components are kind of similar to the original Hinmarsh-Rose model, and because it's a phenomenological model, it's been studied well before to know all the parameter ranges for generating different types of activity. After adding this parameter, we had some insight how to limit the range of variation, the freedom in the parameter space. And then we just use the range for these parameters and use kind of machine learning to just optimize the parameter values.
Do they all have some biological explanation behind them or not yet? Like if you go like enzyme constants and diffusion
Sometimes,
speed and all that.
so in a phenomenological model, it's often difficult to have a direct connection between the physiology and the model parameters. But the most direct connection we can find for like the parameter values and the variables in this phenomenological model is mostly with the time scale of the activity of like different ionic channels and dynamics in the real, you know.
Questions? Okay, all right. Thanks, Amin, again for the presentation.